I'm going to go through some work that has been stewing about in my head for a while, some questions I've been trying to get at, and end up with some of our recent data out of the lab. And I'm going to challenge you, hopefully not with nightmares, but to think about some biochemistry. So how long has it been since you had biochemistry? For two people in here, I know it's been pretty recently because I've had them in class, but I'm guessing for everyone else it's been more than a year. Is that fair to say? Okay. So we'll talk about a little bit of biochemistry, but I'll be sure to tie it back to the cow for you, so no worries there. It goes without saying, and I certainly don't need to tell you folks, that as we go up to the transition period, we're talking about a cow that is a metabolic challenge. She's a metabolic case study for us. And this is a result of decreased feed intake leading up to that transition period, and really the increase in demands associated with the onset of lactation, both in terms of energy and in terms of glucose. So for energy, we know that what is required exceeds what she can consume, and in fact, the mammary gland alone will use all of the energy that she's consuming in the diet post-calving. We also know that the glucose demand of that cow, shown here in partitions, the blue part is the basal requirement of a dry cow. And if we carry that through as she goes through increasing levels of milk production from 25 to 125, we know that there's an increase in the amount of glucose that's required for her to support lactose production. And in our higher producing cows, this ends up being about 25 pounds of sugar a day that have to be made in the liver. And so if we look at that just a slightly different way, when that dry cow that we're talking about, whether she's normal conditioned, average conditioned, or over conditioned, she's going through and she's consuming in yellow the amount of energy that exceeds what's required, the blue line, and so she's in a positive energy balance. Maybe this green line is a little higher, and so she's maybe depositing some body condition. But at any rate, once she enters that the transition period, what she's consuming decreases, requirements quickly increase, and so that cow enters negative energy balance. So what does she do to deal with that? What mechanisms are in place for her to meet that glucose and energy need? If we focus on the liver here, so this is the hepatocyte, and the inner part is the mitochondria, or that energy organelle within the liver cell. And we've got energy-producing cycle, TCA, here at the bottom, and glucose production goes up through the top here through gluconeogenesis. And so normally in a lactating dairy cow or a dry dairy cow, most of the precursors for glucose production are coming from propionate being produced in the rumen. And there's also some contributions from amino acids and then from lactate, although those are much less in percentage or in proportion than propionate. But, like we just discussed, around the time of calving, that propionate supply is decreased as feed intake is reduced, and so she has to have alternative precursors for energy and glucose production. And as you just heard from Rick, that is in the form of mobilized fat presented to the liver as NEFA. And so with that, the glycerol that was the backbone of triglycerides can enter into gluconeogenesis and be used to produce glucose, providing glucose for lactose synthesis. Those fatty acids that were in the triglycerides and are now in the form of non-esterified fatty acids will enter the mitochondria and be broken down into their building blocks, acetyl-CoA units. And that acetyl-CoA can enter the TCA cycle and produce energy. Okay, and this is complete oxidation, if you will. However, what we know is that this cycle, because it is cyclic and requires oxaloacetate, has a capacity. You can think of this in terms of a merry-go-round or a carousel that you would take kids to. For an acetyl-CoA to get onto the TCA cycle, it needs an oxaloacetate carrier. Okay? Just as for a child to get on the carousel, it needs the horse to ride on. So the more oxaloacetate we have, the more acetyl-CoAs can be oxidized completely. However, there is a limit to it. You come to the point where all the horses are full and you have to wait a turn. And so when that happens, there are alternative pathways. One of those is incomplete oxidation, which Rick also mentioned, which leads to the production of ketones. And And we know that when this happens, we see a rise in blood and urine and milk ketone bodies, primarily measured through DHBA. And we can characterize those on farm, and we can decide how many cows should be above a level or a concentration that we think is a cutoff, and try to use that to guide management decisions. But this is what those come from, is that incomplete oxidation. We also know that the other alternative pathway or fate for these acetyl-CoAs is re-esterification into triglycerides that are then stored within the liver. And in many species, humans and rodents especially, these are exported as VLDL. However, we know that that's limited in the ruminant animal, and I'll come back to that more. So what then happens is as the cow goes through the dry period and approaches the transition, we go from an animal that has a very healthy liver. This is a histology slice of liver tissue, so you can see a lot of red, healthy cells there, to at the time of calving having the accumulation of lipids, which we commonly call fatty liver, of course. And you can see that all of these white globules are lipids or triglycerides deposited within the tissue. And so in the U.S. alone, this costs us more than $60 to $70 million a year, and leads to a myriad of comorbidities, reproductive, production, performance, et cetera. And so this definitely presents a set of challenges to us that are part of that transition cow metabolic case study. So what I want to do today is to go through some of this lipid metabolism in a little more detail. We'll talk about mobilization and liver uptake, and we'll talk about esterification and export. And I want to do that so that we can piece together the components or the key players of this. And then I want to talk to you about methyl metabolism. And this is something that's been getting more attention over the last year or two, something I've been focusing on in my research for quite a while. So I want to look at what methyl donors are players in the ruminant animal, the liver's role in that, and then maybe look at some bovine-specific methyl metabolism, what we know and what we still don't know. So just to show a little bit more, highlighting what Rick went through earlier, as the cow approaches the calving interval, we have dry matter intake decreasing, and we get this surge of NEFA here in red. So we get this surge of non-esterified fatty acids coming in to serve as this alternative fuel source, or primary in this physiological state. And we know that there's a correlation between how much NEFA is mobilized and how great the triglyceride accumulates in the liver. So liver triglyceride reflects NEFA mobilization. And this is because hepatic uptake of NEFA reflects two main things. One of those is blood flow, and one of them is blood concentration of NEFA. And so as NEFA increases, in the bottom diagram here, we get an increase in liver triglycerides accumulated that has a very short lag to follow that. Liver uptake accounts for about a quarter of the whole body NEFA flux in lactating cows, and it's similar to what the percent output is from cardiac output or cardiac flow. The other factor that I just mentioned is that it's reflective of blood flow. And what we know about blood flow relative to calving is that the increase in blood flow after calving, so this is 11 days post-calving, compared to about a week pre-calving, is increased twofold. Okay? So we have a doubling of the blood that's presented to the liver. We know that we had a surge or an increase in NEFA in that blood, so the liver is now being presented with more NEFA more rapidly, and that is all taken up by the liver. And so if we quantify that, it has been done, this is some data from Reynolds. At the time of calving, and this is an estimate at the day of calving, compared to pre-calving, NEFA uptake by the liver increases 13-fold. So we've increased blood flow, we increase the NEFA that's in the blood, and in fact the liver does what we think it should, and it takes up that NEFA at a much greater rate. So the question that comes is the liver able to handle that NEFA it's taking up? Is it able to increase complete oxidation, or is it employing these alternative metabolic pathways? And if we look at data from that, this is percent increase, at the day of calving compared to 21 days prior to calving. So this is a relative difference here. And when they looked at the ability of the liver to oxidize palmitate, one of those key triglycerides that's mobilized, there was only about a 10% increase in oxidative capacity of the liver to completely oxidize those triglycerides to energy. What they did find, however, that was very remarkable is that the capacity of the liver to re-esterify those acetyl-CoAs into triglycerides that could then be stored was increased by 188%. Okay? So the liver is able to take up that NEFA and break it down into acetyl-CoA, use what it can use, and then alternatively metabolize the remainder, and then store some as triglycerides. But if you recall back to that diagram I showed, the cow will accumulate triglycerides, but then later is able to use those. So this may represent the storage of triglycerides that could be used later. So what we see from this is the same thing that we've shown you already. Triglycerides are fairly low pre-calving, and as the cow enters the transition period, the triglycerides increase, which we know is associated with that NEFA surge, and with her ability to use NEFA to meet the demands of milk production. So if we go back to our biochemistry diagram here, and we think about the liver in these transition cows, and that triglyceride. So what she can't completely oxidize, she's re-esterifying and storing as triglycerides. And the question becomes, how much of that can the liver handle? And then also, can it be exported or used? If we think about the liver in a healthy lactating dairy cow, that liver weighs about 20 pounds, and it can accumulate a pound of fat by the end of that day she calves. And for every body condition point loss, so maybe those 4 body condition score cows have a lot of body condition they're losing right after calving. For every 1 body condition score point she's losing, that's about 10 to 12 pounds of fat that's presented to the liver with that increased NEFA and that increased blood flow. So certainly we are depositing triglycerides in the liver. Can we help the liver export those triglycerides so that we don't have the symptoms that come with fatty liver syndrome? And that export is through VLDL export in most species. And so just to take a second, if this is a VLDL here, triglycerides are packaged with apolipoproteins, primarily ApoB and ApoE. There's also phospholipids, specifically phosphatidylcholine, which can be generated from choline, and cholesterol. And all of these are packaged together into this VLDL that is exported from the liver and can be used by other tissues in the body. For the dairy cow, that would be the mammary gland of specific interest. But we know that this is not as prevalent in dairy cows as in other species. And so there was quite a bit of work done in an effort or in an attempt to figure out why. And the first place people looked was at the apolipoproteins. Well, it must be a deficiency in those. And so this was some work done looking at apolipoprotein B. Some similar work was done with ApoE, but I'll just use this as an example. And in this work, they compared bovine to rats. And so they looked at the synthesis of the protein itself, ApoB. And as you can see here, bovine in gray, rat in red, there was no difference in the amount of protein that the animal was able to make. Okay. So it's not a deficiency in the protein itself. However, when they used labeled protein to look at VLDL secretion, so how much labeled ApoB was actually secreted from the liver as a VLDL, they observed that the bovine had significantly reduced excretion of VLDL than the rat. Okay? So this wasn't a deficiency in ApoB, and like I mentioned also for ApoE, there's some other limiting factor. So around the same time, there was some work being done trying to develop a rodent model to study human fatty liver. We've all heard non-alcoholic fatty liver disorder, NAFLD, and that's a huge concern in human health. So some work was being done to develop a rodent model to study that. It turns out people are not very fond of having repeat liver biopsies done if they're not necessary. So some work was done where they fed rodents diets that were completely deficient in both choline and methionine. Okay, it's key that this was deficient in both, and I'll come right back to that. And what they found was when they did that, they were able to very consistently induce fatty liver, and on a very timely time course. And the primary way was sometimes beta oxidation was decreased, or complete oxidation, but not always. Sometimes it was staying, but they were able to completely reduce or eliminate VLDL secretion from the liver. So all those triglycerides that were re-esterified and stored remained in the liver tissue. And so this VLDL secretion of liberation was key when they depleted the diets of both choline and methionine. And the reason that is phosphatidylcholine, which I mentioned was a key component of VLDL moieties here, can come from two sources. The first one is from choline from the diet. Okay, so if a human like ourselves eats eggs for breakfast, who had eggs this morning? Well, hopefully somebody had an egg. Did you guys all eat a bagel? We could have a whole nother hour conversation about that. Okay, so if you had eggs, then you got some choline in your breakfast. Choline went to the liver and it was used, or could have been used for phosphatidylcholine synthase. The other way is that the liver can actually make it endogenously from phosphatidylethynolamine. And so this can come from other methyl donors, and we'll look at that a little bit closer here in a minute. It can come from other methyl donors within the liver. So what are these methyl donors? The one we hear about perhaps the most are methionine and choline. Methionine has one methyl group, so if you remember, methyl group is this carbon with 3 hydrogens on it, so it's located on the end of a molecule. So we have a methyl group here in methionine. Choline has 3 of those methyl groups to donate, so these can be given away to other molecules. Betaine, we hear about some in ruminant health, has 3 methyl groups here. And then the one that's most important in human health is folate. We hear about supplementing folate, especially in pregnant women. So these are the methyl donors that are available in the liver tissue. So what are these used for before we come back to VLDL synthesis? They actually have a myriad of uses in the cell or requirements in the cell, and there's some competition between pathways for these methyl donors. Some of these are DNA methylation. We hear about epigenetics, right, on the cover of Science magazine. Prevention of oxidative stress, prevention of apoptosis, or that programmed cell death, where a cell is slated to die. We have energy metabolism and protein synthesis, of course, all being key needs for a methyl group. So of those methyl donors, now we have a list of pathways that are competing for their use within the cell. So I want to take a minute to walk through this pathway. Don't shut off your mind yet, I promise. We'll walk through it together. But this puts together, I think, how some of those methyl donors could interact within the cell. So let's start here at the very bottom where we left off a minute ago with VLDL packaging. And we said that VLDL required phosphatidylcholine, and that it could come from choline from a dietary source. Okay? But that choline also has other prospective or potential uses within the cell. Choline can be used to form betaine, which can donate, remember it has 3 methyl groups can donate, and it can donate those methyl groups to regenerate methionine from homocysteine if methionine is needed within the cell. Okay? Now methionine, whether it's from the diet or it's been regenerated, can donate methyl groups itself. And those are donated through SAM, which is the primary carrier of methyl groups within the cell. SAM can donate methyl groups here coming off to any of the things on that list we just rattled off. Well, one of them pertinent to this discussion would be the generation of phosphatidylethynolamine, which can be used to generate phosphatidylcholine. Okay? So as you can see, there are clear interactions here between these two, and potential ways that these can interact within the same pathways. Now, once the methyl group is donated from SAM, it becomes SAH, which does not have a methyl group and needs to be recharged. It's converted to homocysteine, and that homocysteine can be regenerated to methionine if there's a methyl group available either from betaine or from the tetrahydrofolate pathway, the THF here. Now, either of these two will lead to a downstream production of glutathione. And I'll come back to the glutathione and why we're interested in that in just a second. There's a few things we know about this pathway in ruminants, and quite honestly, there's a lot more that we still don't know, I think. One of those is that about 50% of the methionine requirement can be met through regeneration if needed. Okay, so the cell has a large capacity to regenerate methionine, and that reflects the strong priority to have methionine within the cell as a start codon for protein synthesis or for milk protein. We also know that about 30% of methionine absorbed could be used for choline synthesis if we follow through these pathways. So clearly some potential for interaction of these pathways. The glutathione I told you I would come back to is an antioxidant within the cell. Does anybody take one of those drinks that's an antioxidant drink, or you take a supplement because it says it's an antioxidant, or maybe somebody's read a book like Rick's Neighbor that says that would be good for you? So having antioxidants within the cell is important because it can defer or neutralize these reactive oxygen species that are a result of oxidative stress within the cell. And so we'll come back to that once or twice more, but it is a downstream byproduct of both of these pathways, and so something that's important within the liver cell. All right, so what does this mean at the whole animal level? This is a lot of biochemistry so far, so what? What does this mean for the dairies we're working on, and what does this mean for the producer, for the cow, for that matter? Now, as Kim alluded to, I've done a myriad of sampling and of using models in my career here so far. We don't always know the answer in a cow. It's a very expensive model to look at sometimes. We don't always have the answer yet, so sometimes we have to look at other species. So I'm going to ask you to humor me just a little and look at some rodent data to make up for what we don't yet know in the cow. So one of these studies fed a methyl donor deficient diet to rats that were pregnant and looked at the offspring. So if we think about our lactating dairy cows, the high producing cow that's being rebred, and that fetus is developing, this is something that could be very relevant to us. And I just want to point out a few things that they observed when they depleted the methyl donors. The first one is that in the offspring of the methyl donor deficient fed rat, there was significantly less SAM, that methyl donor. Okay, so we've depleted the SAM, the ability of, or the carrier of the methyl donor. And there was also a decrease in the SAM to SAH, that depleted form, ratio. There was a significant increase in lipid accumulation in the liver, and then also triglycerides accumulated in the liver. Okay. The other thing they did in this was they looked at markers of inflammation within the cell, and so that would be caspase and interleukins. And what they found was that in the offspring of rats fed a deficient diet in the dark blue compared to the control in the light blue, there was an increase in all of these markers of inflammation within the cell, both as a protein and then also activity of that enzyme. Okay? And so what this meant was that there was increased liver inflammation, and we know that that decreases the ability of the liver to make glucose and to make energy. The other thing they looked at was the ability of the cell to completely oxidize fatty acids being presented to it and use those fatty acids to make energy. So earlier I said the acetyl-CoA goes into the TCA cycle and produces energy, and that was a slight oversimplification. It actually produces energy equivalents that then go through the respiratory chain, 1, 2, 3, and 4 main complexes there, and produces energy. So we can measure those complexes as an indicator. And there was, in fact, a decrease in complex 1 and 2, the main rate-limiting complexes, in the offspring of animals fed a donor-deficient diet compared to the control. So now we have increased inflammation in the liver, but we also have a decreased ability of the liver to oxidize fatty acids presented to it. Both of those would be pretty detrimental in the case of the dairy cow. So far, lack of methyl donors in the animal means increased hepatic inflammation, and decreased oxidation. There's one more model I want to share with you, and this is the one that Kim alluded to earlier. There's a couple of different ways. Yes? Would that be carried through the life of the animal? On those, yes. in the rodents, they actually looked through the light, and even at reproductive age, those negative impacts were observed. Yeah, good question. Other questions? Okay, so one of the other ways that you can induce, or some of the other ways you can induce fatty liver in rodents is to feed them a high-fat diet. And we know that that works fairly well. And the other way is to give them alcohol, which induces alcoholic fatty liver disorder. Now, a really quick way to induce fatty liver is to do both at the same time. So I can tell you that from working with mice, you don't have to worry about your toes getting stepped on. You don't have to worry about them swallowing what you're trying to give them an oral drench with or biting your fingers, but you do have to worry about them biting you with their very sharp teeth. They get to be a little perturbed when you're trying to weigh them or measure them. But when you feed them a high-fat diet, they become very complacent. And they're not quite as annoyed with you because they can barely waddle around in the hood. When you also give them alcohol, they are no longer annoyed with you at all. They actually seem rather amused with you while you're trying to weigh them and measure them. And so I am here to attest to you that while I love dairy cows, drunk fat mice are not a bad model to work with. Yes, Rich?
Does that work with people too?
You know, I would like to propose a few case studies for that. Would anybody like to volunteer tonight at dinner? We could have a high-fat diet. Kim, I assume there will be alcohol on this cruise?
Probably.
Some sort? Probably. Okay. So, Rich, maybe you could be my high-fat, high alcohol.
Not a problem.
Okay, great. Can I have my LC at the end? All right, so, sometimes a picture's worth 1,000 words, and that's why I put this histology slide up here. This was a 2x2 factorial, so the top panel here is a high-fat diet with no alcohol. You move to the right, that's the high-fat with alcohol. So you can see the white. It doesn't take a histologist to see that liver slice has a lot of triglyceride accumulation there. The bottom two panels are the same top two treatment combinations with methyl donors. So if you look comparing C to A and then D to B, you can see that the addition of methyl donors really reversed the triglyceride accumulation in the liver and protected the liver from that damage. They quantified all of these things, of course. They found increased SAM when they gave the methyl donors, increased ratio of SAM to SAH, decreased oxidative stress, maintained mitochondrial oxidation, cut off there, and promoted beta oxidation as well. So encouraging oxidation of the fatty acids. Yeah.
What were the methyl donors there that they used in them?
In this one, they put back both methionine, betaine, and then choline. They actually had 3 methyl donor in a complex that they put back into the pellet.
Okay.
Yep. Okay. Any other questions about this one? I'm going to let the picture speak for itself on it. Won't go into too much depth on the individual data. All right, so what does this mean to dairy cows? Where's the status of what we know for dairy cows, why we're here? In the pre-ruminating animal, we know that they have choline and betaine available to them, but as that animal becomes a functioning ruminant, we know that there's a decreased availability of choline and betaine because of rumen fermentation, of course. There's an increase in methyl group synthesis through that tetrahydrofolate pathway. So we're having to employ that endogenous regeneration of those methyl groups. As that ruminant starts to lactate, we add even more challenges to that. Not only are we deficient in choline and betaine, we're deficient in methionine, folic acid, vitamin B. We're decreased on precursors for gluconeogenesis. Remember where we started. We've taken away propionate, our main precursor. So this is an added challenge now. And we have increased methyl group synthesis via that regeneration pathway and through a methionine synthase. So heavily relying on the liver cell to not only now make energy and glucose, but also to regenerate these methyl donors in the absence of enough from a dietary source. And so with this, we think about some questions that ruminating animals we know have low availability of these methyl donors, basic metabolism, so all of that biochemistry and those rodent studies, the drunk fat mice, support the need for methyl donor supplementation, both through direct and indirect effects. And then we also know that there's this VLDL export pathway that we would like to enhance in the dairy cow to avoid fatty liver. And so if we go back to this diagram and think about it in terms of the challenge to the cell, trying to get these triglycerides out while also meeting both energy and glucose requirements. And so at this point, we were left with a lot of unanswered questions. Is inflammation important in dairy cows? It's something we hadn't been able to answer yet. Is oxidative stress, how big of a role does that play in the dairy cow? We hear a lot about it in popular press in humans. What role does that play in dairy cows? Is there an impairment of fatty acid oxidation if we don't have these methyl donors? And are methyl donors sparing? That original pathway I put up showed some interaction, some overlap. Do they really spare each other in the cell, or is there altered priority? And so those were some of the questions that I set out to answer with some work that we've been doing in the lab. And trying to hone in on this balance here, what are the roles and the priorities for these within the cell? And so to do this, we used a cell culture model. And there were several reasons why we did that, but a few of the primary ones is that it allows us to test several different concentrations and combinations at once. Okay, so I'll show you the experimental design in just a minute. For us to do that in an adequately powered cow study would have taken all of our salaries together times a big factor. It eliminates the confounders of other changes, like the hormonal changes associated with the transition to lactation, for example. And so we use primary hepatocytes in culture. This is what the cells look like when they're 4 hours after I've isolated them, and then 24 hours. The picture doesn't show up very well here, but it's a nice single layer of cells that we can expose to different concentrations and different molecules and look at regulation of the cell. So what we did for this was we selected 4 different choline concentrations and 4 different concentrations of VL-methionine. And we did this in order to mimic in vivo concentrations, so what would the liver be exposed to in the cow. So these are based on what's published in the literature. And also concentrations that are equal and then methyl group equivalent. So remember that methionine has 1 methyl group, whereas choline has 3. So being able to select a box in this that would allow comparisons of those. Then we asked the question, does this really mimic the transition cow? And there was one key thing that I was missing here, and that was that NEFA surge. So we replicated that 16 treatment block, and we put 1 millimolar fatty acid cocktail on that, and that was a combination of the fatty acids that are most prevalent in the NEFA being mobilized at calving. Okay? So we ended up with about 32 treatments that we were looking at, and this is just to show you what comes out of this. If we have 4 different concentrations of methionine, so each of these clusters of 4 is a single dose of choline with 4 concentrations of methionine, and we end up with 16. And remember, we also had this with and without fatty acids. But there were no interactions. So what I'm going to show you are main effects only. So we would take for choline 0.03, for example, and group these 4 together. And for methionine 0.016 millimolar, and group this first bar of that cluster, and that cluster, and that cluster. So I'll only show you main effects because the interactions weren't significant. I'm going to come back to what I think that means within the liver. All right, so we start out, let's look at regeneration of methionine. This is just an excerpt from that diagram where homocysteine can be used to make methionine, and that's through this gene, MS, Methionine Synthase. And what we observed was that as we increase the concentration of methionine here in the blue, or increase the concentration of choline in the green, there was no difference in BHMT, the enzyme that catalyzes the conversion from betaine to the downstream, so methyl donation from betaine. But if we looked at methionine synthase, which was the enzyme in the tetrahydrofolate pathway, as we increased methionine concentration, there was a decrease in that enzyme. That made sense. If you're getting the cell more methionine, it's not going to waste energy making more methionine. It has adequate amount. As we increase the amount of choline, however, we saw an increase in the expression of methionine synthase. So choline could serve to donate the methyl group for that regeneration. If we put fatty acids on these cells, okay, and looked at the first one, BHMT, we saw an increase in expression of that enzyme regardless of the concentration of methionine or of choline. And this was really interesting because we know that fatty acids, when mobilized, can regulate gene expression in the liver to encourage oxidation of the fatty acids. What this highlighted for us was that the fatty acids could also increase the use of a pathway that wasn't being used in the absence of the fatty acids. Okay? Now we didn't see the same thing. We saw actually the reverse, that it decreases the expression of Methionine Synthase, okay, the other way of regenerating methionine within the cell. And so if we put these pieces together for this part of the puzzle, when we increase L-methionine, it decreased methionine synthase. Increased choline increased that enzyme, okay, suggesting that choline may serve a role as a methionine, or for the methyl donor in methionine regeneration. And when we imparted a fatty acid load, it increased BHMT but decreased MS. And this highlighted that there may be a preference for the pathway that produces the downstream glutathione. Remember I said we were interested in that as an antioxidant. We know that oxidation of fatty acids does cause oxidative stress, so this was very interesting to us that the fatty acids could regulate a switch or preference for the other pathway in this case. We also looked at methionine conversion to SAM. Remember that key methyl donor, and that's through the enzyme MAT. We didn't see a difference in MAT with either increasing concentrations of D-L-methionine or of choline, but there's also multiple levels of regulation here, and so we're working on looking at protein regulation and then also some other ways of trying to capture that side of the pathway. Okay. We looked at the conversion, the enzyme PEMT that manages the conversion of phosphatidylethanolamine to phosphatidylcholine. Remember I said that that could be an alternative source of phosphatidylcholine for VLDL packaging. And what we found was that there was no change in PEMT with increasing concentrations of D-L-methionine. And so this suggested that the methionine is not being used for that use within the cells. And so one of the things that I had always asked, and I had asked Rick over and over again, is, well, why can't we just say if VLDL is increased or decreased? We often allude to the fact that, and I think Rich will show you some data here later, that VLDL export may have been increased because we see less accumulation of triglycerides, but we've not been able to quantify VLDL. And so this is mainly stemmed from the fact that the lipids in ruminant VLDL are different than those in humans and rodents. And so if you just separate by weight, like we're able to do in rodents and humans, then you don't get an accurate representation of just VLDL, because those techniques weren't made for bovine. And so what we've been able to do, this is very recent accepted abstract data, is a dual antibody-based ELISA, which is a really fancy way of saying we picked a combination of two proteins, and that combination is only present on VLDL, not on LDL or HDL, and so we're able to quantify the VLDL by identifying those proteins, not by the weight of the lipids within them. And so we did that for the samples from this study, and what we found was that there was no difference in VLDL exported from the cell despite the concentration of methionine, D-L-methionine. But as we increased the concentration of choline, we saw a linear increase in the export of VLDL from the cell. So this was very exciting to us, not only because we had validated a tool, and I don't show the validation here, but we had validated a tool for VLDL quantification, but also because this is the first definitive evidence I think we have that says if you increase choline, you can increase VLDL export in a ruminant. The other thing we looked at was oxidative stress, and we did that by looking at reactive oxygen species that could be accumulated. We know that that decreases the hepatocyte function within the cell. The fatty acids, relative to no fatty acids, is how we quantify this. So pairing up those 16 treatments with fatty acids to the 16 without. And as we increase choline concentration, this tended to decrease reactive oxygen species. It was the 2 millimolar dose that had the lowest reactive oxygen species. It wasn't even that highest dose required to see a significant decrease. And there was no difference accordance with DL-methionine on ROS accumulation. So what does this all put together, piece together mean? As we increased DL-methionine, we had decreased endogenous methionine regeneration. The cell had less demand on regenerating endogenous methionine. It did not change PEMT, the enzyme that would be involved in VLDL export, and it did not change VLDL export. And it did not change ROS. As we increased choline, we increased endogenous methionine regeneration across those doses of methionine, tended to decrease reactive oxygen accumulation, and it increased VLDL packaging and export from the cell. All right, so what's this mean to the dairy cow that you guys were looking at yesterday or that we'll be looking at in a couple of days? And I think there are a few key take-homes from this. The first is that it suggests a biological priority or supports a biological priority for methyl donor use. Not all of those methyl donors are used the same way within the cell. And I think that this allows us to look at that there's a whole animal and a cellular level requirement. And I indicated that there was a lack of interaction. And I think that supports this. When we present a cell with methionine, there are so many requirements for methionine, starting as being the start codon for every protein synthesized, and then also for milk protein synthesis. That methionine is being used preferentially for those things, I think. And I think there's more data out to support that. And I'll let Rich and Rick talk about if we see that at the cow level with milk production or milk protein production. But not likely to be involved in VLDL or ROS accumulation as a priority within the cell. Increased NEFA may shift the pathways of methionine regeneration to favor antioxidant production. Cellular methionine can be met by dietary methionine or through endogenous regeneration. And in this study, we saw choline being a methyl donor that could provide that methyl group for regeneration. Okay, so with that, I'd be glad to take any questions or strikes against biochemistry. Either way. Yes.
I have one. The first two questions. One may be very silly. Are there any cells within the body that can metabolize fatty acids except the hepatocyte? I mean, we are always looking to the hepatocyte. There's a lot of fat going all over the place. We concentrate on in the liver. Are there any cells? Is there any other place that the cow can get rid of that fat?
So the mammary gland can use fatty acids to make milk fat.
Yeah.
The liver is the primary source of the primary site of oxidation. The kidney can do a little bit of oxidation and a little bit of gluconeogenesis, but very nominal compared to what the liver can do on a per cell basis, and then also when you multiply it out by the mass of the liver. The muscle can use a little bit, but again, it's very limited, mostly for its own purposes, not for mass oxidation for energy.
Okay.
Yeah.
The other question is, it's clear from your presentation that the importance of choline in exporting VLDLs or packaging those. How about the importance of either of those two in oxidizing fatty acids within the hepatocyte? Because, there are a couple of ways that you can also go to oxidize completely.
Yep.
Have you looked into that pathway?
Not as completely on that side. So the question is, I talked earlier about oxidation of the fatty acids and have we seen differences with those two?
Yeah.
So far we haven't identified differences between the two on changing oxidative capacity.
Yeah.
But again, that's the side of the story we haven't fleshed out quite as much yet. But yeah. It seems that fatty acids themselves are the ones that are able to most regulate the oxidation of the fatty acids. So for regulation of those enzymes, it seems that fatty acids are the key player in regulation, which is interesting in and of itself because they're able to regulate the enzymes that control the oxidation of the cells. So, yeah. Other questions? Yeah, sure.
Yeah, in the cell culture work, I guess maybe what I wasn't clear on was in your media, where you're comparing the different methyl donors, was there an attempt to make sure that that everything else related to protein synthesis, for example, amino acid supply, binding supply, were the basal requirements to satisfy the protein requirements met? Before you start looking at methyl donor.
That's a good question, and I don't go into experimental design real heavy here. But what we do, cell culture media in general is always designed to be in excess of any requirement, so that you can study the one thing you're interested in. So, for example, I make these medias by scratch because the methionine concentration is somewhat like 10 times what we think the requirement is for the liver. And so we design cell culture medias to never have a restricted component unless we want to do that by design. So all other requirements were met in